Welcome, everyone, to the 2026 BioPhotonics Spectroscopy Summit. Thank you all so much for joining us, and in this session, you'll hear from Dieter Bingemann, a chemist and senior application scientist at Wasatch Photonics. Dieter earned his doctorate in chemistry and joins us with 3 decades of research and industry experience in spectroscopy, analytical chemistry, and applied photonics. Today, he'll share how to capture clean Raman spectra for autofluorescent biological samples. If you have any questions or comments, you can type them right into the Summit chat box that is on your screen. And if you missed part of today's sessions, you can always come back later and watch them on demand. That's all from me. Dieter, it's great to see you again, and we appreciate you for taking the time to join us here at the Spectroscopy Summit.
Yeah, thank you so much, Jake, for your kind introduction and for organizing this BioPhotonics Summit and giving me this chance to participate. So, if you're perfectly honest, when you analyze a sample, don't you sometimes wish you could just point a magic wand at your sample and you get your analysis results right away? No more sample prep, no more waiting. If you just caught yourself nodding, then maybe Raman spectroscopy might be something for you, because that's exactly what you do with Raman, like you see in the simple schematic on the left. Yeah, you point the laser at your sample, you collect the scattered light, and turn it into a spectrum. And this Raman spectrum, like the one you see here on the right, contains a lot of detailed information about your sample. In fact, this information is so specific, it's often called a fingerprint. So Raman spectroscopy is indeed an instantaneous chemical analysis without any sample prep. And I would say that's actually pretty cool. So it's almost too good to be true, right? Well, the samples of interest here in this community are biological samples. And traditionally, Raman spectroscopy has been difficult with biological samples because those tend to generate a lot of autofluorescence. And what do I mean with that? Well, in this example of a BSA spectrum, the Raman signals are actually these tiny little peaks on top of this huge mountain of the autofluorescence below it. So I guess you would agree that looks like a little bit of a problem here. So in this webinar, I would like to discuss simple and very practical approaches that you can take to mitigate the strong autofluorescence like you see here for a typical biological sample. And once you do, with these approaches, you too can record clean Raman spectra like the one I'm showing you here for the exact same BSA sample that we've seen on the previous slide. Now, my focus here will be on simple tricks that you can implement easily rather than expensive high-tech solutions. And we will focus on 3 areas: the laser, the imaging, and post-processing. And at the end of this webinar, I promise you too will be able to clean up your Raman spectra just like I did with this one here. Now, for those of you not yet that familiar with Raman, a very brief aside what Raman spectroscopy is. Raman spectroscopy is now almost 100 years old. It was discovered by C.J. Raman in 1928, which earned him the Nobel Prize for Physics in 1930. When light scatters off a sample, the original wavelength of light is scattered in all directions. This is called elastic or also Rayleigh scattering, and it is, for example, the reason why the sky looks blue. In Raman scattering, however, it is in Raman scattering, however, the light does not just bounce off as is. It actually exchanges energy with the sample. And this is therefore called inelastic scattering. The molecule shown here ends up vibration excited, and the scattered light lost is the same amount of energy. That is, the frequency in Raman scattering is now shifted by the vibrational frequency that was excited in the sample. Now, in Raman spectroscopy, the spectrum of this shifted Raman scattering is measured. The spectrum therefore represents the vibrational frequencies of your sample, which is this very specific sample fingerprint that we are interested in. In the most common Raman setup, in the most common setup for Raman spectroscopy, the laser excitation is focused onto the sample with the same lens that also collects the Raman light. A set of mirrors and filters then separates this Raman scattering from any remaining laser light. And the second lens focuses this Raman emission onto the spectrometer. The spectrometer disperses this light into the spectrum that we want to have. Now, the days of do-it-yourself Raman setups covering huge laser tables with nitrogen cooling and hour-long measurement times are fortunately long gone. The technology has evolved dramatically. Today, I want to focus on very compact Raman systems like the ones that Wasatch offers. Shown here on the right is an X-series spectrometer, which has a footprint of the size of a shoebox, basically. It's shown here with an external probe that is fiber coupled to the spectrometer, and the laser is already built right into the instrument. On the left is the much smaller, actually pocket size, fully integrated XS Raman system. These are the exact two spectrometers that I used to generate all the spectra that I'm going to show today. Also, a few words about Raman applications. The application space for Raman is wide and expanding rapidly. Here are just a few examples to illustrate this breadth. Almost all materials exhibit a Raman spectrum, so Raman can provide very fundamental insights into a sample on a molecular level. This is used in basic research in basically all areas, but increasingly also in the material sciences and even nanotechnology, for example, to characterize graphene or diamond-like structures. As I said, Raman also does not require sample prep and the spectra are very specific. So Raman is widely used for sample identification, white powder detection, threat detection, airport screening, but also for incoming and outgoing quality control. And I will later talk much more about the last group of applications in biophotonics. So here just very briefly, that could be food and beverages, process control, biotechnology, or medical diagnostics, but we'll get to that in more detail later. Now, let us get back to the topic of this webinar, which is sample autofluorescence and how to reduce it. So autofluorescence is actually a very general problem for Raman, not just for biological samples. Take, for example, this brandy sample here, another type of sample that has very, very strong fluorescence background as shown by the red spectrum here on the left. The Raman peak we're interested in is circled here on the top. That's the peak of ethanol. And how do we get to this clean spectrum on the right, shown in blue? That again, we'll look at three areas to tweak: the laser, the imaging, and post-processing. So let's dive right in. A common trick to reduce autofluorescence is to switch to a longer laser wavelengths. Shown here are spectra for a glucose sample from a grocery store, so certainly not a super pure sample, and excited with different laser wavelengths, specifically like 532, 638, 785, 830, and 1064, all very common Raman wavelengths. And they're sort of color-coded. And as you can see, the longer the laser wavelengths, like the red 785 or the black 830 spectrum, the lower the fluorescence, compared to example for the 532 spectrum shown in green. For this reason, actually, 785 nanometer has become the most common Raman laser wavelengths to date. Switching to 1064, which is shown in gold all the way on the bottom here, typically removes all the fluorescence. But this switch to 1064 comes at a hefty price. And I don't just mean cost. This longer wavelengths requires a different detector, which is much more expensive, but also has a much higher dark signal, must be cooled to much lower temperatures, and has about 100 times less signal. So this 1064 approach works okay, but it's actually not great. For completeness, I also want to mention two more techniques that come from the laser angle, but they're much more high tech. The first is called shifted excitation Raman difference spectroscopy. And because that is quite a mouthful, it's just abbreviated as SIRS. Not to confuse with SIRS without the D. This is SIRS with the D, or the difference spectroscopy. An example is shown here on the left. The trick here is that one switches quickly between two lasers with slightly different laser wavelengths. And we're talking maybe just half a nanometer like listed here. If you take the difference between these two spectra, you cancel the fluorescence, as shown by the blue curve on the bottom. The disadvantage is that you get higher noise, and you have to deal with two lasers. The other technique is called time-resolved Raman. An example is shown here on the right. Here one uses the fact that Raman is an instantaneous process and fluorescence is slightly delayed on the nanosecond scale. This approach therefore requires picosecond lasers and time-resolved detection, which does not just sound expensive, it really is. So both of these techniques add complexity and cost to the setup. And again, in this webinar, I wanted to focus on simple approaches. So let's turn our attention to the optical imaging of both the laser excitation and the emission collection. Yeah, that's the second trick in our study here. Here I show spectra of a sample of beer. Now, beer also has horrible fluorescence, as you can see. Yeah, in red on top is the spectrum for the beer in a 20 millimeter round vial, like a typical chemistry vial. In blue, the same beer sample, but now in a 10 millimeter square cuvette. Yeah, same spectrometer, same setting, same sample, just a different container. Now, the ethanol signal is marked with a little circle, and it's basically exactly the same in both cases. But the fluorescence background is about 50% higher for the round vial. The round vial seems to act like a cylindrical lens, which corrupts the optical quality and leads to a higher proportion of the fluorescence relative to the Raman signal that is imaged into the spectrometer. So here my point is, even a measure as simple as just changing the sample container can already have a significant impact. So the spectrum shown on the last slide was recorded with an X-series spectrometer system, which has a multi-mode laser. Now we are switching to the pocket-size XS Raman system, which uses a single-mode laser. Single-mode lasers focus more tightly than multi-mode lasers. And here we are tightening this focus even further by switching from the standard lens that's shown here on the left to a 50x microscope objective as shown on the right. So the laser spot now shrinks from about 35 microns to just about 10 microns. Yeah. The sample we study here, you can barely see on the bottom, is a single bacteria colony that grew on an agar saddle plate in a petri dish. And as you can see, the contrast between the Raman signal that's circled and the fluorescence background underneath gets quite a bit better once we focus the laser more tightly. So, and for this bacteria colony, we can actually improve the ratio of the Raman signal to the fluorescence background even further. Yeah. Here we now follow the approach that is typically employed in high-end confocal microscopes. Confocal means that the region that is imaged into the spectrometer is narrowed down with a pinhole. We can actually do the same here with this pocket-sized spectrometer system, just by reducing the detector height over which we read the spectrum. This is a simple software setting, as shown here. Now, the spectrometer really images input to output. So this vertical discrimination on the detector generates a virtual pinhole on the entrance slit, and hence effectively enforces confocal imaging. Now, that is pretty cool. You get virtual confocal imaging with a pocket-sized spectrometer system simply using a software setting. And as you can see, this virtual pinhole actually improves our Raman to fluorescence ratio quite a bit for the same bacteria colony that we've seen before. So tight focus and virtual pinhole, they actually make a huge difference already in our sample here. So let's move on to the third family of tricks that I want to discuss, which is post-processing of the resulting spectra. Now, a common approach for fluorescence removal is called baseline correction. Different methods exist, and most work by fitting a smooth curve guided by some adjustable parameters. Here I'm showing a fairly aggressive baseline correction fit to our bacteria colony confocal spectrum that we've seen before. While this baseline correction clearly removes the fluorescence background, as you see on the bottom here, we are still left with a counting or shot noise that is associated with the higher fluorescence background. The baseline correction can't remove that. So in this example, we followed all the tricks, picked the long excitation, focus tightly, single mode laser, and objective. We used the virtual confocal imaging, and yet many of the small peaks in this baseline corrected spectrum are still hidden in the noise. And another problem that one encounters with highly fluorescent samples, besides that noise, is the so-called etaloning of the detector. This is a common effect in all back-thin scientific CCDs, and it's caused by an interference effect in that back-thin silicon layer, as shown here in the schematic in the top right. This etaloning generates an oscillatory pattern on top of any fluorescence background, which are these tiny wiggles, if you look carefully, on the pure fluorescence spectrum shown on the left. You can remove the background like we did before, but these wiggles will remain as shown on the right. This might look like a Raman spectrum, but it's just the detector's etaloning, and we don't want it. Yeah, but fortunately, etaloning is very consistent, and you can remove it with a pixel by pixel correction specific to the spectrometer. So this etalon correction should therefore always be the first step in post-processing any spectra with autofluorescence. So coming back to our baseline correction, instead of hitting a smooth curve, better ways exist by now for baseline correction. Everybody is talking about AI these days, and for good reasons, because AI is indeed very, very powerful. And with AI, we can remove both the background and also remove the noise. So we have a way to extract clean Raman signals from a noisy autofluorescent spectrum. Wasatch has developed such an AI Raman cleanup tool, and it's based on convolutional layers, which are basically very, very effective filter functions. Here, they are arranged in a so-called UNet. This type of network has traditionally been used for image segmentation, but we have trained a version of it to instead clean up Raman spectra. And we gave the model the quirky name Dalai Raman. This acronym is actually not random. It stands for the network, its origin, and the training approach we used. How does it work? Dalai was trained with tens of thousands of real Raman spectra. Dalai is quantitative. A peak of 1,000 counts hidden in a lot of noise on top of a big background will be recovered as 1,000 counts clean Raman peak. And Dalai is agnostic to the sample. That is trained to recognize Raman peaks in general, not to recognize specific sample spectra. And best of all, it actually works like a charm. Yeah, if I take a 785 nanometer Raman spectrum on a very challenging biological sample, autofluorescent sample, and post-process it with Dalai, the model gives me a fluorescence reduction that rivals that of any 1064 nanometer Raman system, but at the speed and the cost of a silicon Raman system. Well, let's look at a few examples. Yeah, on the top right is the brandy example that I've shown you before. Dalai is applied to the brandy spectrum shown in red, and here it pulls out in blue the ethanol spectrum of the content of the brandy. On the left are spectra for fermentation of glucose with yeast. Yeast adds an enormous background to the Raman spectra, as you can see in the original spectra that are shown on the top right. The color indicates the time over several days how it evolved. But Dalai cleans out this background really nicely, as shown in the middle. Again, you have the color coding of the time. And these results line up really well with the glucose and the ethanol spectrum that are shown on the bottom, with ethanol, as you know, being the product of the fermentation of glucose with yeast. But you also see an additional peak around 1800 here in the middle, which is not in either the glucose or the ethanol spectrum. This is actually a side product of the fermentation, which is carbonic acid. And the bottom right, I show you the poster child of any challenging fluorescent sample, which is sesame oil. Yeah, on the left is the original spectrum, and you can actually barely see the peaks in this fluorescence background. And on the right is the Dalai process spectrum. The spectrum actually looks better than some of the 1064 results I've seen before. And I have to say, I ran the sample on that pocket-sized excess Raman system. So this is pretty powerful stuff. All right, so now you really probably want to know, how does Dalai do on a Raman spectrum for the bacteria colony on a settle plate that you've seen a number of times before? Yeah, again, the spectrum is shown here on the left. I circled the same peak as before that we've been following all along. On the right, you have the Dalai clean spectrum. And I would say that that actually worked out really well. It looks like a pretty clean spectrum all of a sudden. Again, circled is that same peak that we've seen all along. And if you think about it, here we're combining actually all the best tricks in a single measurement. We use the longer 785 nanometer laser excitation. We tightly focus the single mode laser with an objective. We record the emission confocally with a virtual pinhole. We applied etaloning correction, and finally Dali post-processing. And indeed, we get a pretty amazing Raman spectrum for a very challenging sample, bacteria colony sample. So you might ask, is this a reasonable result, right? With AI, you never know. Do these peaks make any sense? So to double check, I added lines at the position of Raman bands that are typically expected for biological samples. These bands are all took from the reference that's cited in the bottom left. And as you can see, the peaks we get in the spectrum and the lines from the literature that are marked with a label, they actually agree really well. And also the magnitudes of the peaks, they actually make a lot of sense. So now you can actually see that the peak we've been following all along is actually for the bacteria colony is actually one of the lipids peak for those bacteria. And best of all of this is really not all that hard to do. You can easily apply this pixel-based ethylene correction and the Dali cleanup just by clicking a few buttons in the Wasatch spectrometer software. So autofluorescence reduction for challenging biological samples, it sounds great, but what could this really be good for? You might ask. So, well, the application space for Raman in biophotonics is actually really big. And to explore this a little further, we will simply follow the three examples that I've shown earlier and pull in similar studies from the literature to expand on it. So let's start with the glucose fermentation. That's actually just the poor man's biotechnology experiments. Raman is already used to monitor feedstock in fermenters to improve quality and yield. Monitoring challenging microorganisms like the yeast we did in our example could now be easier with these fluorescence reduction tools. It could lower the limit of detection, and we could also follow the production of the target and monitor maybe side products that could indicate that the process could be drifting off in unwanted directions. Raman could also indicate the stage of the process, say if the fermentation has already been completed. So autofluorescence could really simplify the analysis and improve the results in biotechnology monitoring. And moving on to the second example, the Beyond Brandy that I showed. We were just talking about fermentation processes. So the same also applies to beer, which is fermented. You can follow the fermentation process, but also the quality control of the final product could benefit from clean Raman spectra for this highly fluorescent and difficult sample type. Natural products that are used in this production often show also high fluorescence of autofluorescence. So performing an incoming quality control on the ingredients can also benefit from this fluorescence reduction. Or looking at the brandy example, it is actually not uncommon for fraudulent liquor to contain toxic levels of methanol. So clean Raman spectra could help to detect this methanol not just in clear liquor, but now also in fraudulent browner whiskeys and brandies. Or drugs are sometimes added to liquor, from spiked drinks to cocaine smuggling. Clean Raman spectra could potentially allow their detection. And finally, some food types are also subject to fraud. Everything expensive, really, like cheese, olive oil, honey. And again, food samples tend to show strong autofluorescence, so fluorescence reduction tools can also help uncover this food fraud. And finally, my third example came from the medical area. We have seen the spectra for bacteria colony in a Petri dish. Now, analyzing bacteria cultures is very common in medicine. So this could be, for example, be a bacteria identification or to test for antimicrobial resistance. The autofluorescence has been a stumbling block in these studies. So fluorescence reduction could make these applications a lot easier. Or tissue analysis, with Raman, it faces the same types of autofluorescence challenges. Raman has been shown to identify cancerous regions, say to check for margins or detect tumors in the first place. And the fluorescence reduction could help here as well. And recently, there's also been a growing interest in disease diagnostics with bodily fluids, all of which show autofluorescence. So cleaning the Raman spectra, as discussed, could potentially advance these medical applications of Raman spectroscopy. The application areas are listed here are some examples taken from the literature, but I hope that this quick overview can give you a feel for the potential of Raman in biophotonics once you add this autofluorescence reduction. So, what did we learn? Raman spectroscopy is a powerful technique. It is information rich and with little sample prep, but Raman on biological samples has traditionally been difficult due to autofluorescence. We looked at three main approaches to reduce this fluorescence: switching the laser to longer wavelength, tighter excitation and smaller observation region, and finally post-processing with using daylight to reduce the fluorescence and noise. We actually combined all of these approaches using the pocket size XS Raman system, and we saw how easy it was to apply these tools. So in my mind, these improvements will really benefit the use of Raman on biological samples. The three examples we looked at were fermentation, bacteria colony, and spectra of beer and brandy. They were just placeholders for a much wider range of applications in the biological and medical fields. And I truly believe that this entire application space can benefit tremendously from the autofluorescence reduction that we just discussed. Thank you very much.
And Dieter, thank you so much for joining us to teach us how to capture clean Raman spectra for autofluorescent biological samples. Before we wrap up here, I just want to remind everyone in the audience, if you haven't already, you can type your questions and comments into the Summit chat box. And do not worry, if you have not been answered yet, you will be reached via email at a later date. This session is hosted by us at Photonics Media, and we certainly hope you're enjoying the BioPhotonics Spectroscopy Summit. Thank you all so much for joining us.